This is Chapter 15, Part 2 video. We're going to talk about the adaptive immune system. The adaptive immune system is a specific response to an antigen. So just to recap, the innate is a non-specific response, so fever, part of that system is triggered whether it's by a cold virus or Ebola, so in a non-specific fashion. Always fast acting and ready to work. The adaptive, you're going to have a specific response to an antigen. If you have a cold virus, antibodies are produced specifically for that virus compared to Ebola, which you would have antibodies produced specifically for that virus. The adaptive immune system is slower, so a primary immune response the first time you come into contact with that antigen takes about 2 weeks for the level of IgG antibody to be at the highest titer in the blood. We're not born with an adaptive immune system. We're born with the ability to produce the adaptive immune cells, and shortly after you are born, that system begins its activities. There are 5 attributes of the adaptive immune system. There's specificity, so that means that any adaptive immune response acts against one particular molecule's shape or antigen and not others. So if you have one B cell become activated, that B cell is going to be specific for one specific antigen. Inducibility, so these cells of the adaptive immune system are activated in response to the specific antigen. Clonality, once the cells are induced, they proliferate, so they reproduce for many generations, and you get a lot of identical cells. Unresponsive to self, which is when our adaptive immune cells act on foreign antigens and they do not act on our own normal body cells. And then memory. Adaptive immune cells will form memory, which accounts for a fast secondary response of that same antigen. So in a secondary immune response, we get antibody produced in about 3 to 4 days on average, rather than about 7 to 14 days for the primary immune response. So the adaptive immune system, this takes place in the organs of the lymphatic system, and it's a surveillance system that screens the tissues of the body for foreign antigens. It's composed of lymphatic vesicles, or vessels, cells, tissues, and organs are all part of this. So again, an antigen is a substance that causes the body to activate the immune system. So it includes various bacterial components, viruses, fungi, protozoa, food, and dust can also be antigenic particles as well. There are 3 types of antigens. There's exogenous antigen or extracellular antigen, so outside of our host cells. So it can be toxins, other bacterial secretions, components of cell walls, cell membranes, flagella. Endogenous antigens, which are intracellular, so when you have a viral infection or intracellular bacterial infections. And then autoantigens, which are self-antigens derived from normal cell processes. In some cases, some of our immune cells need antigen presentation. B cells, for one example, they interact with extracellular or exogenous antigen, but our T cells, they need antigen presentation. So antigen presenting cells will display antigen fragments on the antigen presenting cell surface with what's called an MHC protein, major histocompatibility complex. There are 3 types of antigen presenting cells: macrophages, dendritic cells, which are the scouts, and then B cells are antigen presenting cells. So what we're doing is we're just going through some of the background and introducing all the players involved in the adaptive immune response, and then in class we will actually look at some specific examples and put all the pieces together. This is a dendritic cell. Like I said, they're very important. They're going to crawl around and sample antigen and take those to local lymph nodes for presentation to different adaptive immune cells. The major histocompatibility complex protein is a protein that holds and positions the antigen for presentation. So it's this protein that's produced inside the cell, and it would be packaged with the antigen and displayed on the cell surface. There are two classes of MHC proteins. MHC1, which is found in every nucleated cell, except red blood cells, but red blood cells, they don't have nucleus. And then MHC class 2, which is found on B cells and antigen-presenting cells such as the macrophages and dendritic cells. Looking at B cells, like I mentioned, B cells interact with extracellular or exogenous antigen, so they can interact with free-floating antigen in the body. Their major function is going to be differentiation into plasma cells, which will eventually secrete antibodies. They will also develop memory B cells, which are involved in a fast secondary immune response. B cells have these immunoglobulins on the surface of the cell called B cell receptors, or BCRs. Each B cell, individual B cell, has multiple copies of the same exact receptor. Each B cell generates a single B cell receptor. So, shape. The B cell receptor has a specific shape, variable region to it. There would be about 500,000 copies of that same exact shaped B cell receptor on the surface of the B cell. We produce billions of different B cells, each with their own unique B cell receptor in the human. This would be the B cell membrane here. The B cell receptor, this is a Y-shaped receptor here, and it would have a variable region at the tip. So the antigen binding site here is where it would interact with the actual antigen. So going back to the specificity, this B cell can only interact with this one molecule shape. There would be other B cells that would have different variable regions that would allow them to interact with other antigen shapes. B cells, like I mentioned, they eventually would differentiate after they bind to the antibody or antigen and become activated, undergo clonal expansion, and then eventually differentiate into plasma cells, which secrete immunoglobulins called antibodies. And these antibodies would have that Y shape, just like a B cell receptor, and a specific antigen binding site on that antibody. So the antibody has the Fc stem and a variable region, and that variable region is what attaches to the antigen. So antibodies interact with antigens and in a specific manner. So this antibody has this little triangular shape, so it can attach to the specific antigenic determinant on the bacterial cell. This antibody B here has a different shape, and it can attach to a different antigenic determinant on this bacterial cell. The antibodies produced by plasma cells are different, have different functions based on the type of antibody produced. There are 5 classes of antibodies. There's IgM, which is the first antibody produced, IgG, which is the most common, longest lasting, providing the long-lasting protection, and IgG is what takes about 2 weeks for to build up in your body the highest titer during a primary immune response. IgM, you get a high level IgM after about 7 days, but then quickly diminishes as IgG is going up. IgA is associated with body secretions, provides nursing newborns with some protection if they're breastfed. IgE, this is involved in parasitic infections and allergies, and then IgD, the exact function is not known. Once antibodies are produced and they begin interacting with antigen, there are 5 outcomes of antibody-antigen binding. There's agglutination, opsonization, activation of complement, antibody-dependent cell-mediated cytotoxicity, or ADCC, and then neutralization. Agglutination is basically reducing the number of infectious particles to be dealt with. Since there is a Y shape to that antibody, they can bind to multiple antigens. So here you can see we have 2 bacteria bound together by 1 antibody. Here we have 3 bacteria bound by 2 antibodies. So this reduces the number of times a phagocytic cell has to actually ingest the antigen. It's taking larger bites, basically, compared to single bites of bacteria. It's like using a bigger spoon. Instead of using a teaspoon, it's using a tablespoon or a soup spoon. Opsonization, since that Y shape, that Fc stem of the antibody is pointed out, that causes opsonization or enhanced phagocytosis. So it's an attraction. The phagocytic cell can attach to those Fc stems and gobble up the antigen. You get activation of complement. So the complement is the innate immune response resulting in inflammation, opsonization, and membrane attack complexes and causing lysis of the cell. Antibody-dependent cell-mediated cytotoxicity has to deal with sometimes with large parasitic cells. So antibody-dependent, meaning that the antibodies can attach to a large parasite, such as like a parasitic worm. Cell-mediated, meaning we have cells like eosinophils attached to the antibody. And cytotoxicity, basically we'll get direct cell killing. Perforin and granzyme lytic enzymes are released by the eosinophil, and they would go into the parasitic cells, and they would actually kill the cell from the inside out. And then lastly, neutralization, where we get a complete coating of a virus or bacteria with antibody, and that would prevent that bacteria or virus from attaching to host cells. Also toxin neutralization, where you completely coat the toxin with antibody, so again it can't attach to host cells and cause damage. As a result of neutralization and agglutination, for example, those both lead to opsonization. So most of the time when we have antibodies, we're going to have increased phagocytosis as well. T cells, or T lymphocytes, these cells, they originate in the bone marrow and they mature in the thymus. They have T cell receptors on their cytoplasmic membrane, and the T cell receptors do not recognize antigens directly. They need antigen presentation, and they need antigen presented on an MHC protein. Depending on the type of T cell, they have either CD4 or CD8 protein that interact with the MHC proteins. So there's 2 types of T cells we'll talk about. Helper T cells. These regulate the activities of B cells. They regulate the activities of cytotoxic T cells, which are the other type of T cell that we'll talk about here in a moment. So T helper cells require antigen presentation on MHC class 2. So T helpers need antigen presentation from an antigen presenting cell, and those are the B cells, the macrophages, and the dendritic cells. T helper cells have CD4, and CD4 interacts with MHC2. CD4, it kind of grabs a hold of the MHC2 protein, and it stabilizes the bond between the antigen presenting cell and the actual T helper cell. Cytotoxic T cells, or TC cells, these have T cell receptors. They have a CD8 protein, and CD8 protein interacts with MHC class 1 protein. So what the cytotoxic T cell is going to do is going to be activated by a T helper, and then they leave the lymph nodes and they seek out cells with endogenous antigen. So again, endogenous antigen would be a virally infected cell or a bacterial infected cell. And if you are a host cell that's infected, what you would actually do, you would package viral particles or bacterial particles and display that on your surface. And that way it flags the immune system, it flags our cytotoxic T cells, and lets our immune system know that those cells are infected. So like I said, those cells need to be activated, and then they will leave the lymph node, and then they seek out the endogenous antigen infected cells. They would then produce two chemicals, one called perforin and the other granzyme. Perforin puts pores into the cell surfaces, and then granzyme will actually kill the host cell. So the whole point of cytotoxic T cells are to actually kill your cells that are infected. So if you have a virus, it needs to stop that viral infection by killing it. And again, cytotoxic T cells, they require the antigen presentation on MHC class 1. Now, there are two types of adaptive immune responses. We'll talk about this more specifically in class. There are some worksheets in the course pack that we will do together. The first adaptive immune response is the humoral immune response, which is an immune response to extracellular antigens. It's also referred to as antibody immunity, since antibodies will produce. There's T-dependent and then T-independent, meaning T helper dependent and T helper independent. There's also cell-mediated immune response, which involves intracellular pathogens, so a virus replicating inside of a cell or a bacterial inside of a cell. No antibodies are produced by cell-mediated immune response, but we get direct cell killing by cytotoxic T cells. Now, switching gears to the third portion of the chapter, which would be applications of the immune system. The whole point of the immune system is to gain memory and ward off the antigen. So primary immune response, 3 days, we start to see a little bit of IgM. IgM after 7 days, we get the highest titer, highest amount. As IgM depletes, IgG is building up. After 2 weeks, we get the highest titer. So a primary immune response, we get the antibodies and we get memory cells produced. So an application of the primary immune response is to mimic a primary immune response with a vaccine. So now here with a tetanus vaccine, for example, it's preparing your body for an actual encounter with the tetanus toxin. So we get memory after 2 weeks. So the problem with the tetanus toxin vaccine is that it's not long lasting. So every 10 years or so you need a booster shot to build up that titer of the antibody and to build re-stimulate those immune cells. So with the second exposure, so if you actually came into contact with the tetanus toxin, after 3 days you have IgM, about 4 or 5 days you have a high level of IgG. So that's the whole point of the vaccine. And this would work for all vaccines, mimicking a primary immune response. The benefit of wide mass vaccination programs is we get herd immunity. Since there are certain populations that cannot be vaccinated, so the very young, so infants for some vaccines, they administer them at 2 years of age and beyond. Very old, or some people might be actually allergic to the vaccine. So herd immunity occurs when a large portion of the population is vaccinated against a disease. So we have a lack of susceptible hosts, and we have an inability of the virus or bacteria to actually spread. So this is responsible for the dramatic decline of many childhood diseases. And as we know, right now in Michigan, we have a measles outbreak which is due to a portion of the population that was not vaccinated for the measles virus. This is a figure showing the introduction of two different vaccines for the polio virus. So in the 1950s, 1960s, the 1950s, we have the first virus vaccine introduced, and before that we would have 40,000 cases in the United States each year. Introduce the first vaccine and we see a dramatic decline, 5 to 10,000 cases, and then we have a better vaccine that's introduced in the mid-1960s, and we basically eradicate polio from the United States. The problem with the polio vaccine at that time was it would have residual virulence. So 1 out of every 2 million people would actually get clinical polio from the vaccine. Since the year 2000, we've switched to a different vaccine again. It's a inactivated vaccine where there is no risk of residual virulence. Measles vaccine in the 1950s, 1960s, we would have 700, 800,000 cases of measles each year. Introduce the vaccine, and again we get a dramatic decline of the measles virus. Measles was pretty endemic, and we see a large decline again in the 1980s with more and more vaccines, and now we're starting to see an uptick in the number of cases again because people are not vaccinated. There are three main types of vaccines. There's attenuated vaccines, which are live pathogens with reduced virulence. So I like to say they're a tamed down version of the actual virus. And this results in a mild infection. So the measles, mumps, rubella, that's an attenuated vaccine where you get an active immune response, a strong immune response because of an actual infection that is occurring. One of the benefits of attenuated vaccines is that it can provide contact immunity, and it can actually infect others around them. So even if you're not vaccinated, but you're near somebody that was vaccinated recently, that infection can spread to you, and then you would actually be protected as well. Caution must be warranted because the modified microbe might retain enough residual virulence to cause disease. So pregnant women, HIV patients need to be cautioned if given an attenuated vaccine. There are inactivated vaccines, which come in two different forms: whole agent, which is a deactivated entire microbe, and then subunit vaccine, which is just fragments or bits and pieces of the microbe. Both are safer than live vaccines. There is no infection, but due to a lack of replication of the microbe, the fragments don't produce as strong as an immune response, so booster shots would often be needed. And then the last type of vaccine would be toxoid vaccines, which are chemically or thermally modified toxins that stimulate the adaptive immune system. So these are useful for some bacterial toxins such as tetanus and diphtheria. And again, these would require multiple doses due to few antigenic determinants, a very weak immune response, basically. There are vaccines against certain diseases other than the childhood diseases, anthrax, cholera, plague, tuberculosis. However, those are limited to special populations such as military and laboratory personnel. The last topic here would be vaccine safety, and there are common problems associated with immunization. It can be mild toxicity, which is the most common, where you get pain and fever from the actual immunization. There's risk of anaphylactic shock because the way we produce vaccines are often in chick embryos. So if you're allergic to eggs, the egg proteins or preservatives can cause allergic reaction. And like I mentioned, the residual virulence from attenuated viruses occasionally causing the actual disease. And then lastly, the accusations that vaccines cause autism, diabetes, and asthma. There is no cause and effect relationship showing evidence for the causation of these diseases from receiving a vaccine. Research has not substantiated those allegations. This concludes the Chapter 15 video.